Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. I'm really excited to be joined today by Dr. Brent Richards, who's professor of medicine and human genetics at McGill University and CEO of 5' Sciences. Brent is a physician scientist who spent his career using genetics to figure out what risk factors actually cause disease, and he's done some amazing work both in endocrinology, infectious disease, and also applying human genetics methods to drug discovery more broadly. So we're going to try to cover a bit of ground today, and Brent, I really appreciate you taking the time to join me.
Yeah, thank you, Patrick. I am very happy to be here. I'm a fan of the podcast. It's been really fun to watch this podcast evolve and also have the opportunity to learn a lot about what's going on in people's minds that we don't often see in papers through the interviews that you conduct.
Yeah, well, I appreciate that. And actually, you probably know one of the topics that we come to a lot is genetics and drug discovery. And one of the things that's always been on my mind is how we have big pharma companies like AstraZeneca, Regeneron, Amgen that historically have huge infrastructure and have invested significantly in genetics. But then we have everybody else that needs to make sense of the enormous amount of genetic data and the implications that it has on their pipeline. And one of the topics in constantly in my mind and with some guests is actually how do the playbooks and opportunities differ, whether you're a small biotech, mid-size, you're at scale. Some of the resources like UK Biobank are democratizing, but also there are just some things that you can't do everything when you're a tiny company. And it struck me that that's probably a great place where you all fit in, and maybe a reason you exist is that every company to some extent needs to consider genetics in the drug discovery process, but not every company has that capability. So let's start there, and maybe you can help me with the premise and a little bit of why you are doing this.
Yeah, so I think that all of this journey starts with a personal mission. So I got really excited about human genetics as an undergrad, and then really have tried to focus my career on being able to use that tool to improve the care of my patients, and never really started out with an intent to make a company which would help with drug development. But as you pointed out, it's just become increasingly useful, and to the point that I see often in investment committees that people want to check off the box and say, like, does this have human genetics support this drug development program? Because most of the people know around the table that this will increase the probability of success importantly. But the problem is that checkbox opportunity doesn't really play out uniformly, because there's different types of human genetic evidence that can support a drug development program. There's different types of data, single cell, there's GWAS, XWAS that can support that program. And helping the teams actually translate that information into a target profile package that allows them to think about the therapeutic opportunity and bring that back to improving patient care has been a really rewarding adventure for us at 5'.
You talked a little bit about different types of evidence, and probably not all types of evidence are equal. Do you have a framework or a sense of the hierarchy or pyramids of evidence? Because people will be familiar with tools like Mendelian randomization and genome-wide association studies and other more maybe mechanistic cellular assays or otherwise. How do you think about what types of genetic evidence you bring to bear in drug development?
Yeah, so I mean, as you know, all of them have limitations and all of them have strengths. And the trick is to be able to triangulate across them so that the different types of biases from the different types of study designs become orthogonal to each other. And so if you end up getting the same answer from different points of view, despite those orthogonal biases, then you probably have a much more probable drug development program that's going to succeed. We do in the field has generated some evidence that some types of information are more useful than others. GWAS is really sensitive, right? So if small perturbations of that genome can shine through GWAS because the sample sizes are very large. And so if you see no signal at all in GWAS at a very large sample in a well-conducted study with a good phenotype relevant to your drug development program, that's a signal that maybe there's something that you want to think twice about. And there's obviously many exceptions to that. So if you look at the SGLT2 locus for type 2 diabetes, which is a really successful drug target that I use to treat my patients with diabetes, you don't see much of a signal for diabetes, but yet it's really useful in the clinic. And we know that people who have Mendelian loss of function variants in the target of SGLT2s, they have glucosuria, which is not a phenotype that we study in the clinic in large-scale genetic studies. So proteogenomic Mendelian randomization is a really useful tool because we can get a causal biomarker, we can get a direction of effect, we can get a circulating biomarker that can be used. Sometimes we know the tissue from where it's coming from. And then, of course, XWAS is like the crown jewel of information, but it's very rare that we actually have enough coding genetic variation to be able to tie a gene confidently to a disease. And very few of the successful drug development programs that are supported by human genetics actually have XWAS. That's prompted some players in the industry, as you pointed out, to invest billions of dollars in XWAS information. Hit rate's really low, though. So you have a whole spectrum of evidence that you have to think about, synthesize, triangulate, and then bring that to that investment committee to try to understand, is this something that's going to have wheels and get us to the clinic?
It sounds like one of the things you see very regularly is maybe someone has arrived at a target that they want to go after. And then the question is, how much genetic support is there, or what's the nature of the genetic support for this target? But then you probably also have the novel target discovery in the first place, right? And that's where the XWAS can be used. So I'm thinking about the Regeneron paper that came out relatively recently, maybe like last couple of weeks. It'd be good if you could talk a couple of recent examples like that to just help people put these in some categories in their minds.
Yeah, so lots of different categories. So we often end up in a novel dark biology space. So we're humans, we're social creatures. We like to do things that are being reflected in the people in our community who are doing similar things. This is not unique to genetics, not unique to drug development. You look at finance. If you're on the edge of a cliff and you got to pull the trigger on a several $100 million investment, you want to look along that cliff and see that your colleagues are there pulling a similar trigger, right? Michael Lewis brings this up in Moneyball, which is a great look at the psychology of investment. We feel very confident making bets if we're going to jump off that cliff with some of our peers. And we've actually looked and quantified this in 5 parts. So we tried to understand what are the successful drug development programs and what are the determinants of successful drug development. And when you think about that for a little bit, Patrick, you're actually trying to solve 2 problems. So you're trying to solve which cliffs are people going to walk up to, and which cliffs are they going to jump off and have a success.
Right.
And then the most important determinant of finding a cliff that you're going to walk up to and actually do that clinical trial and get the money to do it, is just simply the number of papers that are published on the target. It has an enormous effect. The odds ratio is 5. So if you want to get a drug development program started, look for a target for which we know a lot, or at least we think we know a lot. But guess what the effect of that broad literature coverage is on approval status?
I'm going to guess, I'm going to guess maybe 1.3 odds ratio or something meaningful, but not massive.
It's even worse than that. It's null.
Oh, geez.
So it's 1.0 with tight confidence intervals around the null. So we as a community are stuck in this trap of having to make high risk bets with a high degree of failure going after a novel target's really hard. So you saw that with a really nice triglyceride to HDL ratio paper that was published last week from Regeneron as well as the academic community. Totally novel target. To walk up to that cliff and get somebody to write you a several $100 million check is difficult. And we're stuck in that psychology as a community. So one of our missions at 5 Prime is to help people engage that ambiguity, to understand that being on the edge of a cliff with your peers is maybe not where you want to be.
Yeah.
Yeah.
What do you make of I'm putting you on the spot on this one, and we can cut it if not, but there's another trial that jumps to mind about the Zeus IL-6 inhibition trial. This was a pretty high profile failure a couple of weeks ago, and my memory is my understanding of the literature is a little sparse on this, but my understanding was it had pretty good Mendelian randomization evidence behind the target, but maybe not a huge odds ratio. And in any case, the trial failed very definitively. It was very well powered, and I'm wondering what you make of cases like that where actually you have really good genetics evidence up front, and then what do we learn from those where people have jumped off the cliff and not landed it, so to speak?
Yeah. Yeah, it's hard to stick the landing sometimes, even when you have confidence going in. So IL-6 is a tough one. So Novo Nordisk, as you know, in the Zeus trial, targeted IL-6. And one of the problems with signaling cytokines is you can have really good evidence from Mendelian randomization, and often that comes from the receptor. So let's say you have a genetic perturbation that increases the resistance to signaling from that cytokine. That genetic perturbation then drives up the cytokine and then can influence the outcome. So you can have then different results whether or not you look at genetic determinants of the cytokine that are cis versus at the receptor. Because remember, if you're driving up the signaling cytokine because you're inhibiting the signaling, that cytokine can influence other receptors that you might not know about.
Right.
And so you actually may be seeing that signal not because you're dampening signaling through IL-6, but you're actually activating IL-6 driven pathways that are not through the IL-6 receptor because you have more IL-6.
Right.
So in our academic lab, we actually have a paper that we are finishing now showing examples that are in cytokines where we actually see this effect that secondary signaling drives a reverse signal than what you would expect given the known pharmacology of the gene. And so even with really powerful tools like proteogenomic Mendelian randomization, you can get the answer directionally opposite to the truth.
Wow.
So this is why we say, you go to an investment committee and somebody says, is there human genetic evidence to spread this? Yes. Okay, move on to the next question. That's not a very good place to land. You want to have a really good understanding of what are the sources of bias in this tool set so that you're not just checking a box and moving on to the next question about pharmacokinetics of the target.
Yeah. So to the extent that it's possible to be prescriptive, what is a really gold standard package around a target look like? Because you mentioned we can have a ton of literature and that will help you get the sign off, but it actually doesn't influence the success rate at all. So there must be something hidden in there that's not really about volume, but it's about specific quality. And I imagine it's going to be really different per program. But are there some essential pieces in there that do make something actually have a lot of downstream predictability of success when you're going to go after the target?
Yeah, there's some, and again, there's tons of exceptions, right? And so it's really hard to be prescriptive about make sure 1, 2, 3 are done, and if they're done, then you're fine, because that's not the case. So these are really things like GWAS, XWAS, Mendelian randomization, knowing what the causal gene is at the GWAS locus. We've spent a lot of time at 5 Prime working on the genetic determinants of progression versus susceptibility.
Yeah.
So as you know, the most of genetics is based upon susceptibility, which is obviously useful. But when somebody comes into my clinic, I'm not very often trying to prevent a disease. I'm often trying to treat it. And we know from work from Adil Harood, who is a neurologist at McGill now, but did his postdoc, where he showed that the genetic determinants of MS susceptibility and progression are totally orthogonal.
Really? Huh.
Yeah, so that was published in Nature. It did a great job. He went through that painstaking process of collecting data from people who had multiple sclerosis, following their outcomes, adjusting for the drugs, because remember, these people are treated, so their outcome might be entirely influenced upon by the drug. And then you have to know when they took the drug and for how long and what was the dose. And so it's a really hard problem to solve. But Adil was able to show not very many targets that influence progression, but that they're entirely different in the case of multiple sclerosis from those that influence susceptibility. And so we've spent a lot of time at 5' developing methods that actually can allow us to be able to use simpler ways to identify those progressions. So putting together that package, synthesizing it in a way that is translatable to the other colleagues around the table is really hard. Actually, something at ASHG we're going to have a symposium on, which people can visit our website and sign up for, where Robert Plenge is giving a keynote on how do you translate human genetic evidence into decisions. Mark McCarthy will give a talk from Genentech, Sally John from BMS, and Jim Travascus from Lilly, talking about really the difficulty of what you're alluding to. How do you make a decision that's probabilistic with a lot of unknown unknowns?
Yeah.
Yeah.
Yeah. And do you have a sense of how, because you have this investment inversion as well, right? Where you are going to make a big investment on this, but it takes time and money to chase down some of these questions early. So I imagine a lot of the questions about what of these unknowns, how much risk can we tolerate in this process, right? Can we actually wrap our arms around what risk we are taking and understand what that is? So at least if we have a negative result, we learn something, right? From and don't we don't end up with an underdetermined equation that we can't really solve. Is that
Yeah.
a good way to think about it?
Yeah, I think it's a very good way to think about it. Exactly. And all of these things are learning experiences, unfortunately, for the field and for our patients. But we have learned a lot from them, and we can modify investments. So people in teams are making a lot of investments in AI right now, very appropriately. But almost all of that investment is going into making new molecules, right? So most AI focused drug development is based on making new molecules. That's not actually the most important problem. That's not the single most important determinant of failure. It's just that we gave the drug in the trial, it engaged with the target, and nothing happened to the patient. It's lack of efficacy that plagues the fact that when I go into my clinic, a lot of times I say, there's not much we can do to help you. And that lack of efficacy is most often just driven by the fact that we're picking targets that don't cause the disease in humans. They look great in the model system. They look great in observational epidemiology. But when we drugged it, nothing happened.
Yeah.
So
what's the biggest data gap behind that? Because we have, you described a lot of methods that we can apply, but ultimately it's only as good as the underlying data. What are the key things that are missing to close that phase 2 failure gap that I think you're talking about?
Yeah, so we have a lot more data now, as you know, but the systematization of analysis of that data and building a pipeline across a portfolio that yields systematic results that are comparable across targets is very much missing. We're going to get a lot of boost in performance, I think, over the next few years as we explore non-European genomes. Not because those genetic variants have different effects in different populations, they almost always have the same effect. But the thing that we're going to identify is a lot of genetic variation in humans that we've never been able to see before.
Yeah.
Models are models. Associations are not causality. And understanding that with human genetics, which is a sharp tool to be able to actually see what's happening in humans without reliance upon a model system and being able to make a claim of causality, can help to solve a lot of those efficacy problems. But we think in human genetics, everybody's doing this in industry. So you talked about Regeneron, AstraZeneca, GSK, they're doing a great job. But we are helping a lot of the teams that are just starting to think about these tools, starting to think about making these investments. And that's the vast majority of the industry. So I think the biggest impact will be just systematically developing the ability to do this across a pipeline, de-risking portfolios and getting more medicines to patients.
Yeah, that makes total sense. Is there some, yeah, I think you all have I'd like to actually talk about the products that you've built. I think you have one called Primex that does something like you describe. But also like how your journey into this, because I mentioned this at the start of the episode. Your I asked you how many feet you're in 5', how many feet you're in McGill, and I think it probably requires more feet than you have. But I would love to just talk a little bit actually about what got you interested in doing this in the first place, and stepping out of your academic role to put this hat on as well, and then I'd love to hear a little bit about how you balance or split the time between the two, and how they feed into each other, because I was saying this before we started the recording, but I believe more than ever academia and industry are closer and closer together. You have people moving back and forth, you have hybrid type of roles like you're doing. So I just really want to learn a little bit about how you made the decision, and how you spend your time.
Yeah, yeah. So, I mean, making the decision was pretty easy once I thought about it. The My personal mission has really been very clear from when I started the career, is just to try to use human genetics to improve the care of my patients. So I got excited about that during my undergrad. And then when I got to medical school, I forgot entirely about research. I thought, well, that's incredibly boring. I want to, get my hands dirty. I want to like do brain surgery. I want to deliver babies. And, research was like the last thing on my mind until I got a little bit more senior in my training. And I remember one morning walking into a patient's room, and I'm an endocrinologist, so I was on the endocrinology service, and this man was crying, and it was early in the morning, and we were prepping for surgery, and he was about 65, he was going to have his second below knee amputation. And he just said, look, is there anything else you can do with me? I really want to be able to play soccer with my grandson. And he said, my diabetes has actually been pretty well controlled. Like, what is it that you're not understanding about my disease? Because I'm not going to have feet at the end of the day. And you guys are missing what it is that you should be treating in me. And I thought, he's entirely right, because back in that time, so that was in the early 2000s, we were treating diabetes by just giving insulin and pushing glucose into people's cells. And then people in industry and academia thought, oh, well, actually, maybe we could get it to dump into urine through SGLT2 inhibition, or maybe we could get people to stop eating so much food through GLP-1 receptor agonism. And those are great genetically validated drug targets. And so I had absolutely no role in either of those in either of those interventions, but it's been probably 15 years since I've been involved in the amputation of a foot or a leg of one of my patients. And that's because we actually have been able to use these tools to intervene on physiologic processes. And it actually works, which is amazing, because if you look at the Manhattan plot for type 2 diabetes, there are so many loci. And to think that I haven't sent a patient for an amputation in approximately 10 to 15 years, because we're taking down a few of those skyscrapers on the Manhattan plot with 2 drugs. So it's working.
Yes.
And so when I saw that, and we wrote a paper in 2012 on, do we recover the drug targets of osteoporosis using just the GWAS of bone mineral density? We wrote that in Nature Reviews Genetics, and the answer was yes, every single one of them. And so it became to germinate in my mind, okay, maybe this could be useful for drug development. We got increasing calls from industry to be able to help them. And then during COVID, I realized that this is an opportunity for us to be able to contribute to a lot of the companies that don't have these resources. And if successful, maybe we could actually realize this mission. And then the second question that you asked is, how do you do academia and industry work at the same time? Because that's a very good question. And I think a lot of your listeners have either been in one camp or the other, or have been committed to one and think the other one's ridiculous, or there's so many different opinions across the spectrum of this. I've just been really fortunate that I've had great teams inside academia as well as inside 5', really capable people, been able to make the investment in them. And you can make totally different investments in skills and capabilities inside industry. And then you go to academia, and it's like carte blanche. You can do whatever you want. So you have different constraints. You can invest for the long term in the industry. You can build the pipelines. You can build the data sets. You can offer stable jobs to people that are well paid, which has obvious advantages. And when you go to academia, I can work on things that, like, for example, trying to understand the differences in genetic determinants by sex, which is probably not too relevant to drug development, but very relevant for what it means to be human.
Yeah.
And that's a very privileged place to be in. And really, it's been achieved by having the good fortune of having the right people to work with.
Yeah, it's amazing. I appreciate that, and I think maybe it's something we shouldn't take for granted. I had a PhD supervisor that was very supportive of industry,
Yeah.
and it definitely shaped the direction I went. And I had friends who didn't have that kind of environment, saw it as a little bit of a one lane or the other,
Yeah.
and it can change the way you think about the world. And yeah, we should keep that in mind, and give our students ways to get outside of whatever box that they're in, right?
Yeah,
And broaden
exactly.
horizons. Yeah.
Yeah. Because, there's nothing, we're all trying to help patients, right? So there's nothing that I give in my clinic that hasn't required both,
Yeah.
right? Every single intervention that I'm able to give somebody to help them, there is an element of academia and there's an element of industry. So, to try to paint everything with, we don't need this other team. This is a ridiculous. It's a false choice because
Yes.
we're all trying to achieve the same thing. We just have different incentives, different motivations, and different limitations in doing so. And if we can embrace those and everybody play their role, then we get to be able to help patients.
Yeah, absolutely. With the endocrinology theme, continuing on that lens, I would love to get your perspective on how you see the obesity and GLP-1, but broader target landscape playing out. I've been following the literature a little bit. There's obviously an enormous raft of clinical trials going after dual agonists, triple agonists, and they're incredibly successful. There's also a challenge, though, of the amount of weight loss that's coming from muscle, and so there are other targets like Inhibin E that Wave Life
Yeah.
Sciences I know is pursuing. There may be others as well. There are new targets like FNIP1 that Regeneron and other colleagues just published on. So there's more targets coming into the pipeline. I would just love your perspective on how you see that evolving over time, because it's one of the biggest, it's the biggest market right now, and the patient impact is already enormous, and the potential is even bigger. So I'd love to have your perspective on how you think that might play out.
Yeah, so great question and a difficult one. The It's really exciting. So I think about it from my clinical point of view, and then I'll tell you about what I think about it from like a drug development point of view. From a clinical point of view, it's amazing. So to have a gentleman walk into your clinic and say, "Wow, I went for the first run in the past that I've had in the past 40 years because I've lost 200 pounds."
Wow.
Like, that's amazing. And people are, I have patients who've been able to become pregnant, who've been trying for years. So the clinical impact of GLP-1 agonism is amazing. And now we are moving on to dual agonism and triple agonism. Things will continue to get better for our patients. The drug development scene is really exciting because we have lots of companies investing a lot of money. Obesity used to be the graveyard of drug development, right? So if you wanted to ruin your career, then you should go work in obesity drug development because you'll never achieve anything. But thankfully, some people, particularly in academia many years ago, stuck at it. And industry has spent years being able to develop tools to be able to assess. But now the investment in the pipeline and five prime supports Eli Lilly in the space of obesity drug development. So we've had the opportunity to be able to identify really cool novel targets, and Eli Lilly has the opportunity to be able to work upon those targets. And so the tools that we have now were almost unbelievable even 5 years ago. So I really see an acceleration in this space. We will, of course, asymptote, right? We're not going to be able to make everybody be of like the healthiest weight because there's only so many interventions that we can give. But what I really see is the ability to give multiple interventions at once. as well as being able to solve some of the problems that we have in our clinic, which involve muscle loss, which involves some of the nausea and vomiting that we're seeing. But remember, a lot of the patients at my hospital, a third of the inpatients have type 2 diabetes. So that's like remarkable. Most of those people have type 2 diabetes because they're obese, and that we have a long way to go to get those BMIs down to acceptable weights that we're keeping a large proportion out of the hospital. So great to see lots of teams working on this, and really exciting to see some of the early phase clinical trials that are coming out.
I wanted to ask you about another topic that I find really interesting, which is genetic modifiers and how important we talked a lot about actually how do you select the right target in the first place, but it's maybe related to the point you made about genetics related to progression. We can also lump in genetics related to treatment response.
Yeah.
I saw a paper, this is probably 2-3 months ago now, where they looked at retrospectively at the SGLT2 in one of the SGLT2 inhibitor trials. I think it was Declare, and they looked for people who were carrying monogenic cardiomyopathy variants, but presumably didn't know about it. They were enrolled in a type 2 diabetes trial, but they were carriers of a rare and very damaging cardiomyopathy risk variant. They found the SGLT2 inhibitor, if you stratified by that genotype, it was extremely, it had an extreme positive impact on preventing heart failure in those carriers. And there's all sorts of interesting examples like that where we're hopeful that randomization splits those across our arms for smaller trials. That hope may not be a reasonable plan. And I just love to have your perspective on how much people are thinking about that, how seriously people should be thinking about those hidden genetic confounders that might make their way into smaller trials.
Yeah, so good question. And one that I think is really salient right now, because lots of people are designing such trials to be able to use genetics for recruitment. And you can actually stumble into a real hornet's nest of bias that we have. We actually have an upcoming platform at the ASHG where Matt Tudball, who's one of the lead scientists at 5 will describe. But you may remember the reanalysis of the PCSK9 trials from Amgen and Regeneron, where we saw that individuals at the highest polygenic risk for coronary artery disease had really amplified effects of PCSK9 inhibition, not dissimilar to the experience that you're sharing with SGLT2 inhibition. What we realized in working with Liz Cerulli at Helix and other scientists is that those reanalysis of those trials are very plagued by collider bias. So let's walk through this because it's a little bit complicated. Yeah. Maybe I'll just even talk about what collider bias is, because it's not.
Please.
It's not something that maybe a lot of your listeners. So George A.V. Smith, one of my mentors, has a great analogy to be able to describe this. So collider bias is, let's imagine that you're on an American college campus and it's elite and it's hard to get into. And you can either get an academic entrance or you can get a sports scholarship. Okay. So just imagine, it's not hard to imagine that university probably exists, and some of your listeners have probably spent.
Yeah, I think I went to one. Yeah.
Or maybe they worked there. So you're on that campus, and if you walk up to one of the students and you ask them, "Oh, well, are you great at soccer or hockey or long jump?" and they say, "Oh no, I'm a horrible athlete." Well, then you can infer that they're probably a good student. Yeah. And so by taking the general population and sub-selecting to 2 paths to get into that college, you induce a negative correlation between athleticism and academic success that you don't see in the general population. Okay? So that's an intuitive way to describe collider biases. When you make a subpopulation with different ways to get into that subpopulation, you either get in through door A or door B. Okay. So now let's move that.
Great analogy.
Yeah,
I really like that.
George, because it's incredibly complicated. I have to draw a diagram every time I think about collider biases.
I like that better than the variables. I normally visualize that graph where you've got.
Yeah, that's right. The diagram, and your head's going to explode, and which way does the arrow go? So I just like George's analogy because it's simple for simple-minded people like me. Okay. So if you take like the Fourier or Odyssey trials, which are PCSK9 inhibition, the way into those trials is that you had to have prior coronary artery disease or high risk of coronary artery disease. And you do that because you don't want to treat the general population because it takes way too long to do the study. Okay. So there's different doorways into that trial, and you can get into that trial because you have a high polygenic risk, or you could have a high non-genetic risk.
Yeah.
Right? And then if you're in that subpopulation, just like the American College, you have a negative correlation between those two factors. And so Matt has been able to show that this could explain a large degree of the effect that we're observing of the polygenic high-risk individuals, that a lot of the very large effect upon outcomes could be driven by collider bias. And that's very dangerous because at my institution, we were planning a clinical trial to actually do just this. And randomization does not fix this, right? You can randomize who goes to your American college, but if there's only 2 doors to get into that college, then you're going to have the exact same problem. And polygenic risk scores and other genetic factors can be really helpful to be able to identify a population that's going to have a higher probability of an event. Therefore, you have an enriched population for the outcome. Therefore, you can make your trial smaller and more focused. But then you also run into this collider bias problem, which you can show in observational data. You can show in randomized controlled trials. And as a clinician, we're always like, oh, bias is bad. Well, just randomize it and everything will be fine. But randomization does not solve this. And so we really need to think carefully when we're reanalyzing trials or designing prospective trials that this type of bias can greatly increase the magnitude of the effect that we see, which is very often spurious. That doesn't mean that it's always spurious, and it doesn't mean that we can't use genetic risk to be able to identify a population that's going to have an event, but we have to be really careful about that interaction that you were referring to.
Yeah, and I guess this any trial that is maybe I'm going out limb here, but any trial is going to have one or more of these doors you can get into, right? If it has a genetic background, you could have, if it's genetics and environment, you have a genetics group and an environment group. How do you solve for that issue?
Yeah, so you can use in observational studies, you can use inverse probability weighting, which actually works quite well. But it's going to take some thought by the community. It's not like there are hundreds of trials that are being run on polygenic risk scores now, and a lot of that is being done for business development reasons, like you don't want to fragment your population. But there are definitely some, and this is something that's happening both in academia as well as industry, and so we really need to touch the brakes, think about these problems before we commit patients to medicines or even de-intensification of therapy because they're in one polygenic risk strata or genetic risk strata, which we've seen from a trial doesn't need an intervention.
Now, does a fresh trial that is where the polygenic score where polygenic score, for example, is the inclusion criteria, does that suffer from the same issue, or if the way you get in the trial is interesting? Yeah. So why is that?
Yeah, just because. So let's say you get into the trial by having a high risk or a prior event, like in the case of coronary artery disease, is very easy to document. Did the person have a heart attack or not? So there are both non-genetic and genetic ways to have that event. So if you're taking your population subsetting to individuals who only have had such an event, there are some people who got there because of their genetics, some people who got there because of the environment, some people got there, a lot of them who got there because of both, but you induce a negative correlation between the two. And so that is why it's problematic. We don't think a lot of people in the community are aware of that, so we're just trying to spread the message that it's something that we really should think carefully through.
Yeah, but what if you just said, okay, this is a trial for people with high polygenic risk? Is that a cleaner design?
Yeah, so that is cleaner, because that is your trial entry. But then you have invariability in the polygenic risk. I mean, to some degree, right? But you'll have less variability in the polygenic risk, and therefore the trial results, as we always try to remind our residents in clinical medicine, are applicable to the types of patients who could have made it into the trial.
Yes.
And then that becomes difficult from a business development point of view, because if you're going to treat the top 10% of the population, then that means that 90% of patients are not eligible for your medicine. Right?
Yeah, this is fascinating.
Yeah.
For people who are listening who are interested in this kind of field, do you have any suggestions on where they can start to wrap their minds around? Not, a lot of people will be familiar with the basics here, Mendelian randomization, GWAS, but to start to get into some of these deeper concepts, where do you recommend people go to learn more?
Yeah, so good. I mean, we're going to be talking about all this at our symposium at the ASAG, so people are welcome to join us there. The problem is that's like 1 hour, these things have taken a slow-minded person like me like decades to learn. So what I do is try to surround myself with smarter people, learn from them, but importantly, read the literature, because a lot of these things do actually show up in the literature. A lot of people have spent the time and horrendous effort that it takes to get a paper published to be able to share those learnings with the community. It does work. But one thing that I have done, and probably a lot of your listeners have realized, is that reading a paper that's a bit outside your field is hard, right? Because all this jargon. But who's great at that is an LLM. Yeah. So you can, I do this all the time. I was on holidays last week. I just uploaded a paper that I was interested in into an LLM, and I don't understand this, explain it to me. And that's a really great way for people who struggle, as I do, with these concepts can try to understand what's going on and learn from other people in the field.
Yeah, I do the same. I remember when I was starting my PhD, I got some of my supervisor's most recent papers, and I would just read a sentence. I didn't understand it. I'd Google as much as I could until I understood that, and then I move on to the next sentence, and it's a little bit like that.
It's a little bit painful, yeah. Yes, like reading a tax report or something, yeah.
Yeah, but then you learn the words over time. Brent, this was amazing. I really learned a ton, and I'd love to do this again sometime. Maybe the answer to my own question was we'll go into some of these in a little bit more detail, because I think these kinds of questions are worth a lot. Patient impact, time, wasted resources, and if you can get these right while you're designing or thinking through a study design, it can really make a big difference. So yeah, I appreciate you sharing this with us.
Yeah, I really appreciate the opportunity to be on the podcast, and I've really enjoyed learning from the other people. So keep going, because this is an excellent forum for us to learn from each other and eventually try to do what we're all doing, which is improve the lives of our patients.
That's right. Well, thank you. I really appreciate the kind words. And everybody, as always, I appreciate you taking the time to listen, and we'll see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.